Thanks for this opportunity to talk, Byron, and thanks for organizing this meeting again this year. I don't have any disclosures to make related to this talk. And I just want to acknowledge all the great work that the Orthopedic Rehab Association has done over the years, founded in 1990. And education and research are the cornerstones of the efforts to enable us to continually progress in our efforts to advocate on behalf of our patients. So I think we've seen that in some of the presentations given already, and hopefully I'm able to reflect some of that in the talk here. I want to talk about peripheral nerve surgery and some of the current things that are going on in that. I'll briefly review just some anatomy and classification in case people need a little help with the language, and talk briefly about nerve transfers because they've really been an advance in helping our patients recover over the last 10 or 15 years from some of these nerve injuries. But then to focus on some research updates, these are some things that we've been working on here, talking about nerve recovery when there's an associated fracture, when you have both fracture and nerve injury, some of the time sensitivity that we know exists, but some new information related to this. We're trying to develop some synthetic nerve graft conduits that I'll share about a little bit. And then some really exciting stuff about blocking Wallerian degeneration using polyethylene glycol and some associated techniques. We've known about nerve injuries for centuries. Homer wrote about it in the 8th century BC when Teokuk was in a battle and he got hit in the brachial plexus and he dropped his bow. But then he actually recovered and got back into the fight. So in hindsight, we knew we had what we would characterize as a neuropraxia injury. So we've known about these for centuries. And, here's some anatomy from Da Vinci. And then we've learned more about the anatomy too. We know these primary, the anterior and posterior rami exist, the plexus exists, these peripheral nerves then run down to the muscles and motor end plates exist. And then they transition to the muscles that drive the joints. And then, nerves are not simple structures. They're vascular elements, arteries, veins, and then these layers, the epineurium, perineurium, endoneurium, Schwann cells, axons, myelin. Here's a little H&E view, a little higher magnification of that. So we call these nerves, but there are all these sort of structures that are wrapped together, and all these elements are part of injury and recovery. And this basic classification scheme still holds up pretty well, where the neuropraxias are the more minor ones that recover pretty quickly. Those are the ones that you want to have. And then there's a neurotmesis injury, which is the more severe one, which are the ones you don't want to have, where generally all the structures are injured, the myelin, the axon, the endoneurium, perineurium, and the epineurium. And then there's this in-between crowd, the axonotmesis, where some of the structures are injured, some are not, and you may get recovery, but you may not. But that's the basic language. And the neurotmesis injuries are the ones that I'm going to basically talk about here, and that most of the attention is paid to, the higher grade injuries. And one of the great things for these higher grade injuries are these nerve transfers. And people have been talking about these for about 15 or 20 years, but they're just increasing in usage and our skill at doing these and our timing for patients. And so I just want to advocate for looking out for patients where this still can, has an expanding role. Here's an example of somebody that had sort of a C7, C8 type injury where they can take out their ulnar nerve. And we can now bypass part of the median nerve into the ulnar nerve and reanimate these ulnar muscles. So this is done relatively commonly now for ulnar neuropathy and really is a powerful tool to help our patients. Now, obviously, there's concern about donor morbidity. Can you take parts of the median nerve and not have loss? And we've gotten better and better at that. So the answer is yes, you can take parts of the median nerve and reanimate the ulnar nerve. Here's where some of this work started with what we call the Oberlin Transfer, where you can take part of the ulnar nerve and transfer it into the muscocutaneous nerve. Here's a muscocutaneous nerve that's not working. And you splice out part of the ulnar nerve into that to reanimate the bicep muscle. And so this is just a really powerful tool to deal with some of these peripheral nerve injuries, or even more central nerve injuries too. Spinal cord injury, these have roles. And if you can't take the ulnar nerve, you can take intercostals and swing those over. So there are donor choices that you have. And so I just want to talk about that briefly, because I think that's a really powerful tool that we did not used to have. And now we're doing with relative frequency with really pretty good success. I want to transition to talk a little about some of the research that we're doing with peripheral nerves. One of the projects that we undertook about 4 or 5 years ago that has always been an interest to me is when you have a fracture with an associated nerve injury. Here, for example, is a humerus fracture, and there's a radial nerve injury associated with this sometimes. We talk about what's the best way to treat the fracture, but we haven't really focused on what's the best way to treat the nerve. And generally the thought is treat the nerve non-surgically. Unfortunately, most of them recover either from a neuropraxia injury or an axonotmesis low-grade injury, but not always. And so we decided to focus on the nerve. What's the best course of action in treating the nerve injury? So we designed a study looking at this in rats, where we created sciatic nerve injuries in rats in their legs and then broke their femurs. And in half of them, we fixed their femurs, and the other half, we didn't fix their femurs. And then we looked at the nerve specifically to see in which group did the nerves recover better, in the ORIF group or the non-ORIF group. And we looked at electrophysiology, we looked at nerve conduction, force generation, and we did some histology work too. So there are some of the co-investigators on this project that I just want to acknowledge. And this is what the femurs look like. In the ORIF group, we used 2.0 plates. They were 4 holes, and they all healed in the fracture group, in the ORIF group. In the non-ORIF group, none of them healed. They all went on to these fibrous non-unions. And we looked at 36 rats in this at 2, 4, 6, and 8, and 12 weeks. And what we found, to summarize, was that the nerves healed better in the ORIF group. The nerve conduction velocities were better. The force generation in the anterior tibialis muscle was better. We looked at the histology. Here's an ORIF nerve. This is a non-ORIF nerve. The cross-sectional area was better. The nerves looked healthier. The axon counts were higher in the ORIF group as well. The axon counts didn't quite reach statistical significance, so that's why I'm not significant here. But the cross-sectional areas were better. And in general, just macroscopically, they look better too. So our electrophysiology, our histology both supported better nerve recovery with ORIF, which was our hypothesis, that rigid internal fixation of a fracture promotes superior recovery of a concomitant peripheral nerve injury. And we're trying to get this published right now. We hope it receives favorable reviews. It's been presented at a number of meetings where it's been well reviewed. I want to talk a little bit about the time sensitivity of these nerve injuries, because once you have a nerve injury, a cascade of events starts to happen where things happen in the peripheral nerve, things happen in the motor end plates, things happen in the muscle, and some of these things can be non-recoverable. So once a nerve injury happens, you activate a cascade of events where you have a limited time frame in which to work. And might we be able to understand this time frame better and then treat our patients better? This cascade of events was recognized by Waller, multiple decades ago. Back in 1851, he described the degeneration that occurs after a nerve fiber is severed, and it's named after him, Wallerian degeneration, which we all know. But we've come to know a lot more about this, particularly in the last couple of years. There have been a couple of groups in China that have done a molecular analysis of what's happening in the peripheral nerve after a high-grade nerve injury. This was published in 2017, looking at rat sciatic nerves at multiple time points after a non-recoverable nerve injury, doing mRNA analysis, looking at what molecules are expressed in the nerve. And then just in the following year, another group out of China in 2018, a separate group, did the same thing with muscle. They looked in a sciatic nerve injury model in rats at 1, 2, 4, 8 weeks, looking at the molecules that are expressed based on mRNA activity to describe what happens as a result of Wallerian degeneration. So we're learning more about this, and I'm going to reference back to these in just a bit as well. We're trying to build nerve graft conduits right now too, where we're trying to combine a hydrogel and nanofiber composite where we can mesh these things together and then rebuild nerve graft defects with these synthesized materials that can be on the shelf and loaded potentially with things related to nerve healing. And we're trying to get an NIH grant for this right now. This is a pilot study to support the application, but here's a 1 week of our conduit, and here's a 3 week, just showing that neural elements can grow down these conduits. And this was a bit of proof of concept to show that we can grow nerve tissue down these hydrogel nanofiber conduits, hopefully to support this NIH application. And then our strategy is to take these synthetic conduits and then load them with certain things. We can load them with cells, and we can create a gradient to promote healing down these conduits. And our hope is to begin to think about loading them with specific molecules that can be directed by some of this mRNA information. So that's our strategy with the nerve graft conduits. And another thing that's been exciting recently related to recovery of peripheral nerves is the fact now that we can block Wallerian degeneration. I think I can say that as a fact based on multiple studies over the last 20 years, really. In 1985, this researcher, George Bittner, published this article talking about reconnecting severed nerve ends using polyethylene glycol and blocking Wallerian degeneration, immediately having continuity of the axoplasmic tissue and then conducting action potentials immediately after a neurotmesis type injury. And then he's continued to publish on this for the last 20 years. Here he is again writing in 2016 about the curious ability of PEG fusion technologies to restore lost behaviors after these nerves are cut. So this is known to actually block Wallerian degeneration. And people call it PEG fusion or application of PEG, but it's really a three-step process where I've shown the process here in these figures where the nerve is cut, and then an influx of calcium is thought to contribute to the triggering of this Wallerian degeneration. So a primer solution is used to wash out the calcium, and a calcium-free solution is washing this out. And then you use an antioxidant, methylene blue. You reapproximate the nerve ends, whether through suture or maybe tissue. That's another thing we're looking at. But then applying this PEG, the PEG fuses the tissue, and then you can immediately conduct down the pathway. So 3 steps: calcium-free solution lavage, methylene blue, antioxidant, and then the PEG fusion. And this has been done in humans overseas. This has been done in digital nerves by this group here and published. So this is published now, showing a superior outcome to traditional suturing technique in these digital nerves. To my knowledge, it's not approved in our country through the FDA, but I think it's soon to come for mixed nerves and motor nerves in particular. So this is coming, and we wanted to understand, does the suture technique make a difference in how effective the PEG is? Because when you start messing around with this, you start to wonder, does a tighter suture repair matter? Does a looser suture repair matter? When you're not using PEG, it doesn't seem to matter, either a loose or tight repair. In fact, a looser repair with less foreign body may actually be better, but with these fusions, does more suture help or does it not help? And so we did a small study, again, in rat sciatic nerves, looking 2 versus 5 stitches in these repairs. And we really found that there was no difference in immediate conduction, velocity, or force generation in the anterior tibialis muscle. And so either 2 or 5, much like non-PEG neuroorrhaphy, doesn't seem to make a difference. So I think that empowers the surgeon to use 2, 3, 4, 5 sutures, depending on what's necessary biomechanically. And again, this has been submitted for publication. Hopefully it's viewed favorably. These are some of the collaborators people have done this work with. And then I began to think about, well, what about RNA expression and molecular expression downstream from a PEG fusion? And so we got some funding and got a grant and got IACUC approval for this. And this was done prior to those publications I referenced before from China, which it was interesting to me that they were kind of thinking about the same thing. But I was thinking about mRNA expression after PEG fusion, both in the nerve and the muscle, to see if we can start to identify what genes get coded for, what gets upregulated and downregulated. And so just recently, we've submitted this one where we're looking at gene expression both in muscle and nerve after PEG fusion at 24 hours and 4 weeks later in a peripheral nerve injury, a sciatic nerve injury in a rat model. And this is kind of a summary that at 24 hours, we saw all sorts of activity in the tibialis muscle in terms of gene activity, molecular expression. At 4 weeks, it was very quiescent. In the sciatic nerve, there was almost no activity at 24 hours downstream for the injury, but then substantial activity at 4 weeks. And the fact that those 2 groups out of China published their articles without PEG, we could go back and corroborate our control group back to them. And our results are very similar in our control group, looking at what they published in terms of gene expression. And then in terms of the pathways that are activated, you start to get these really complicated things that really only people that are sophisticated with RNA and genetics can look at this. This is beyond my understanding, really, but there's tremendous significance. These were p-values to the -13th, -12th, -10th, in things that are expressed in the PEG nerve downstream versus the non-PEG nerve downstream. And similar levels of significance were expressed in the anterior tibialis muscle downstream. These were p-values to the -11th, -10th. So we're starting to identify what molecules are expressed, what gene pathways are activated, with the idea then that we can modulate or manipulate these things maybe in a systemic way maybe in a preloaded nerve graft that's used to amplify the effects of PEG, or really maybe even be able to do this without PEG, but somehow to combine this information, things we're learning from PEG, things we're learning related to this gene expression as a result of PEG application. So PEG application clearly has a molecular impact. There's differential gene expression, and we're trying to understand the key molecules, because I put a list of things up there, but they may all be connected to certain pathways that are triggered by just a couple molecules. And so here's an acknowledgement of some of the people that have been working on with this one. And, as I just mentioned, then the hope is to kind of bring some of these ideas together, to have synthetic nerve graft conduits, perhaps in these gap models, where we can take the lessons from PEG and the RNA molecules that are expressed, maybe cell cultures, and load these nanofiber hydrogel composites to enhance the healing of peripheral nerve injuries when we can't achieve primary neuropathy. If we can achieve a primary repair, then perhaps we can use the PEG and use these systemic considerations to modulate the downstream activity that's occurring in nerves and muscle to amplify the effects of PEG and further block Wallerian degeneration. So I appreciate the time to talk about these things. None of these things have actually been published. They've been presented at some national meetings, and reviews have been favorable, and I'm hoping that they achieve publication soon. They're all sort of out in submission processes. But I've talked about the work looking at enhanced nerve recovery in a stable fracture environment. I've talked about some of the time sensitivity and things that are expressed relative to Wallerian degeneration in both distal nerve and distal muscle, both with and without PEG. And then the hopes that some of these synthetic nerve graft conduits may have a role in treating our patients with these peripheral nerve injuries, either with a gap or without a gap. And it's just really exciting to me to think that this thing that we've really accepted for centuries now, that Wallerian degeneration is going to happen, that we now have some tools at our disposal and some information at our disposal to move in a direction where we can actually block Wallerian degeneration. So thanks again for the opportunity to talk. I just want to acknowledge, my admiration for all the people that have worked in this rehab world in the past, and publicly acknowledge my time again with Marianne and the influence of Ranchos in her career, and indirectly then to mine. And then also Dr. Perry, who's no longer with us, but I think lives in the hearts and minds of all of us connected with this rehab work and the Rehab Association. So thanks for this opportunity to present. And if there are questions, I'm happy to entertain them.
Questions?
So how much is pain a limiting factor after the nerve surgery?
I'm sorry, I couldn't quite hear the question.
How much is pain a limiting factor after your surgery?
With these nerve transfers, I found the recoveries to be fairly easy pain-wise. Most of these, I can't say all of them, but a lot of these surgeries are getting done as outpatients. A spinal accessory in the shoulder is an outpatient procedure. An Oberlin ulnar nerve is an outpatient. An AIN is an outpatient. So I haven't seen it really trigger CRPS or significant pain for these. And we're starting to do them more often now, even in compressive neuropathies. For example, if somebody has a really bad cubital tunnel syndrome and you see this atrophy distally, they'll get a cubital tunnel release plus or minus a transposition, and then an AIN transfer distally, all as an outpatient. So I really haven't seen pain as being a significant factor in the postoperative care and rehab of these patients. And then the more severe injuries, the spinal cord injuries, where we're doing these nerve transfers, that a lot of these patients don't have tremendous sensory activity anyways because of their losses. And so pain's not a huge issue for them. And I should mention too, one of my corroborators in the this PEG stuff and RNA stuff is a guy that has focus on epilepsy and CNS stuff. And we're thinking we may be able to even modulate the environment for spinal cord injury or CNS injury to reduce some of these peripheral nerve losses and after injury. So that's a long answer to your question, but I haven't found pain to be a huge issue in the recovery of most of these patients.
We do have a question from the Zoom chat. Harry, if your audio is working, you can go ahead and ask your question. Otherwise, I'll read it for you. So he asked, is it possible to stimulate a nerve after PEG and have a motor response?
Yeah, absolutely. That's in fact how we confirm that we have a PEG fusion, because the application of PEG is a little fickle, and when you starting to work with it, you really don't know if you've affected a fusion with PEG or not. And so what we do is we do the nerve repair with PEG, and then we stimulate above, and you can see immediate muscle contracture downstream after the PEG fusion. Now, we've learned a little bit about stimulation because you can stimulate at a certain level where you overdrive the repair even without PEG. So in a lot of these studies in the lab, we'll use some kind of intermediate threshold that's just a little bit above a normal stimulation threshold. Say, for example, I can stimulate with 1 volt and get a muscle signal distally. I know if I do a primary nerve repair, I can stimulate with 10 volts and just drive through that repair. Somehow the signal just goes through the tissue or the fluid around that nerve repair. But if I stimulate with 3 or 4 volts, that won't drive through my nerve repair, but it will stimulate through an effective PEG repair. So we use that to confirm that we have an effective PEG repair is immediate contracture downstream with stimulation above the repair, but stimulation with a sub-maximal level so that we don't overdrive just the traditional repair. And then these rats, they're walking the next day. They're dorsiflexing and walking. It's incredible.
An announcement. If you are in the audience and want CME, you have to sign the CME roster in the lobby before lunch.